DNA replication.
By us.
What makes it up? There are many enzymes that are involved in DNA replication, and we're about to name them all for you.
Really?
The first is helicase. Helicase is responsible for unwinding the DNA double helix, breaking up the two strands. Now we have single-strand binding proteins. These proteins will make sure that these two strands are kept separate from each other.
Cool.
And we have primase as well. Primase will build primer. You're asking what primer is, and it is where DNA polymerase will know where to start. And now we have topoisomerase. Topo what? Just know that it is responsible for making sure that the DNA double helix does not break while DNA replication is happening. And now we have some more terms to know. Yay! The first is the origin of replication. It is where helicase will start from. Now we have the leading strand. This is the faster of the two strands, and it's because of the direction it goes in. And we also have the lagging strand. This is the slower of the two strands and is made up of Okazaki fragments. Now we have anti-parallel. Anti-parallel simply means that while it's parallel, it goes in the opposite directions. By directions, we mean 5' and 3'. These are directions that DNA can go in. It is based on their backbone of carbon. And we have Okazaki fragments. Okazaki fragments are just simply parts of the lagging strand that are not complete. And here's our DNA double helix. Yay!
Woo!
As you can see, there is the point of origin. And as you're about to see, there is a helicase! It's about to unwind the DNA double helix leading to two strands. As you can see, there is also topoisomerase, making sure those two strands don't break. And here you have single strand binding proteins, making sure that our two strands are kept separate. Yay! Thank you! And now we have primase and primer. This is where the DNA polymerase will know where to start. As you can see, the red is the lagging strand and the blue is the leading strand. And there is DNA polymerase 3. It is adding nucleotides to the leading strand and completing our DNA. Yay!
Yay!
Now, here's the lagging strand, the slow one. It is adding some nucleotides called Okazaki fragments, but these fragments are all broken up. That is, until we have DNA polymerase 1 show up. These will remove the primer and just make the fragments a little bit more complete. But DNA ligase will finally mend it all and make it one continuous strand. And yay! Now we have our two DNA double helix. This is called semi-conservative, as it's one part original and one part new. Yay! Bye.